So today we are going to know about this chapter biotechnology and application from this NCERT. So biotechnology is use of living organism, cell or biological system to develop product or process for specific use, especially in medicine, agriculture and industry. In agriculture, genetic modified crops that are we call it GM crops. For example, Bt cotton. It consists the Bacillus thuringiensis gene to kill the bollworms. And golden rice. It is rich in vitamin A. It consists the beta carotene gene added. Then there is herbicide resistance gene. Example, Roundup Ready soya bean. Then pest resistant and drought resistant varieties are present. In medicine, genetically engineered drug, for example, the insulin, Humulin is produced in the E.coli, and growth hormone, interferon, and vaccine. And other topic present in this chapter is gene therapy, replicating defective gene. Example for SCID and ADA deficiency. We will know about it, gene therapy, and molecular diagnosis for that PCR, ELISA, DNA fingerprinting is used. I had made a video on DNA fingerprinting, you can watch it. Then for macrogenomics, personalized medicine using genetic data, that is the for macrogenomics. Then industrial applications for they are the biofuels, bioplastics, enzyme in detergent and food industry. So biofuel, ethanol from sugarcane, biodiesel from algae, then bioplastic made from the microbial fermentation. Enzyme in detergents is amylase and protease. Then in bioremediation, I have made a video on bioremediation, you can watch that. In using microbes to clean oil splits and heavy metal. Biomining, extracting of metal using a bacteria. And waste management, methanogenesis and biogas and plants. So biosafety is important, testing the GMO for allergies and toxicity. Then bioethics, we can check the crops if they have any allergy or toxicity. For that biosafety is done. Then bioethics is moral questioning about the cloning, patenting life forms. Then transgenic organism, biopiracy and bioinformatics. These are important terms in this which you have to learn. So what is transgenic organism? Organism with the foreign DNA inserted. Biopiracy is exploiting the native resources and knowledge without permission. It is biopiracy. That is bioinformatics, using computer tools to analyze biological data. One example, there are various things in bioinformatics which we will discuss. There are databases and all. We will make a different video on that. Then quick mnemonic for application, agriculture, industry and medicine and environment. All these field biotechnology is used. So let's know in detail this chapter. So in this chapter, the Green Revolution succeeded in tripling the food supply, but yet it was not enough to feed the growing human population. Increasing yield have partly been due to the use of improved crop variety, but mainly due to the use of better management practices and use of agrochemical fertilizers and pesticide. For farmer in the developing world, agrochemicals are often too expensive, and further increase in yield with existing varieties are not possible using conventional breeding, as a traditional breeding technique failed to keep the pace with demand and to provide sufficient, fast and efficient system for crop improvement. Another technology called tissue culture got developed. So what does the tissue culture mean? It was learned by the scientists during 1950s that whole plant could be regenerated from an explant, and that is any part of the plant taken out and grown in a test tube under sterile condition in a specialized neutral medium. This capacity to generate a whole plant from any cell explant, it is called totipotency. So it is important to stress here the nutrition medium must provide a carbon source such as sucrose and also inorganic salts, vitamins, amino acid and growth regulators like oxygen and cytokinin etc. By applications of these methods, it is possible to achieve propagation of large number of plants in a very short duration. And this method of producing thousands of plants through tissue culture is called micropropagation. Each of these plants will be genetically identical to the original plant from which they were grown, and that is they are soma clones. Many important food plants like tomato, banana, apple, etc. have been produced on commercial scale using this method. Try to visit a tissue culture laboratory for better understanding. I have made a video on tissue culture, plant tissue culture. You can visit it, watch it. So another important application of method in the recovery of healthy plant for disease plant. Even if a plant is infected with a virus, the meristem, that is the apical or axillary, is free from the viruses. Hence, one can remove the meristem and grow it in vitro to obtain a virus-free plant. Scientists have succeeded in culturing meristem of bananas, sugarcane, and potato. So, scientists have even isolated single cell from a plant, and after digesting their cell wall, have been able to isolate protoplasm that is surrounded by the plasma membrane. Isolated protoplasm from two different varieties of plant, each having a desirable character, can be fused to get a hybrid protoplast, and which can be further grown to form a new plant. And these hybrids are called somatic hybrids, while the process is called somatic hybridization. Somatic cell hybridization, also known as the protoplast fusion, is a technique in plant biotechnology where two different plant cells with their cell walls are fused to form a hybrid cell which combine the trait of both parent plant. So here, isolation of protoplast. A plant cell has a cell wall. The enzyme like cellulose or pectin are used to digest the cell wall, and this give a protoplasm of a plant cell with a cell membrane, that is plasma lamella, and no cell wall. So selection of two parent protoplasm. You select protoplasm A and B here, and fusion is done. Protoplasms are fused using a chemical like PEG, polyethyl glycol, or electric current or electron fusion, and the two cells stick together and their cell membrane fuses, and it is known as cell fusion. Then this is the formation of synkaryon. After the cytoplasmic fusion, their nucleus are also fused, and the fused nucleus is called synkaryon. Then that result in the hybrid cell, somatic cell hybridization, with combined genetic material from both the parent plant, and this hybrid cell can be grown in a culture to regenerate a new hybrid plant. So combine useful trait like disease resistance plus high yield for a different plant and create hybrid between the species that can't normally breed. So important in crop improvement, especially in tomato, potato, and tobacco. So cell wall gonna fuse and clone. Remove wall, fuse cell, merge nuclei, and grow hybrid. If you imagine a situation when a protoplast of tomato is fused with that of the potato, then they are growing to form a new hybrid plant. This combining the potato and tomato well can be achieved, resulting in the form of pomato. Unfortunately, this plant does not have all the desirable combinations of characteristics for its commercial utilization. There can be such plants and obtained, and plant, bacteria, fungi, and animal whose genes have been altered by manipulation are called genetically modified organism, GMO, or GM plants have been useful in many ways. These are made crops more tolerant to abiotics, for example, cold stress, drought stress, salt tolerance, then heat stress, and reduced reliance on chemical pesticide. Pest resistance crop are made. It helps to reduce the post-harvest losses. It increases the efficiency of the mineral usage by the plant, and this prevents earlier fertility of the soil. It enhances nutritional value of the food. Example, golden rice. Example is vitamin A in the enriched rice. In addition to this, GM has been genetically modified crops has been used to create plant to supply alternative source to industry in a form of starch, fuel and pharmaceuticals. Some of these applications in agriculture that you will study in detail, that is decreasing the amount of pest by using the Bt toxin, is produced by the bacteria called Bacillus thuringiensis, Bt for short, and this Bt toxins gene have been cloned from the bacteria and has been expressed in plant to provide resistance to insect without the need of insecticide. In effect created a biopesticide. Example are Bt cotton, then Bt corn, rice, tomato, potato and soya bean. Bt cotton, some strain of Bacillus thuringiensis produce protein that kills certain insect. Now these insects such as lepidopterans, tobacco. So here this image tells about the mechanism of action of Bt toxin. Bacillus thuringiensis is an insect pest which is a core concept of agriculture biotechnology, especially the Bt crop like Bt cotton. First is the insect, usually a larva or a pest like a caterpillar, eat the plant containing Bt toxin crystal protein. These are protoxin that are inactive in the plant. Then inside the insect gut, which is an alkaline in pH, the crystal dissolve and proteolase in the gut activate the protoxin into an active toxin. So this process is known as the dissolution of the protein crystal and activation. Then the activated Bt binds to the specific receptor on the epithelial cell of the midgut of the insect, and this is highly selective process. The toxin works only on an insect that have these receptors. So binding to the specific receptor, and then vacuolation, cytoplasmic leakage and cell disruption takes place. Their binding lead to the pore formation in the gut cell membrane, and this cause ion imbalance, vacuolation, leaking of the cytoplasm and eventually cell lysis. The insect dies due to the gut paralysis and septicemia, entry of the gut microbes into the body cavity. Now how you will learn this Bt toxin action? First step is ingestion of the toxin, second is dissolution and activation, then binding to the receptor and vacuolation and cell disruption. Budworm and armyworm. Then coleopterans, they are the beetles, and dipterans are the mosquitoes and flies. The Bacillus thuringiensis form protein crystal during a particular phase of their growth, and these crystals contain a toxic insecticidal protein. Then why does this toxin does not kill the bacillus? This Bt toxin protein exists as an inactive protoxin, but once an insect injects the inactive toxin, it converts into an active form of the toxin due to alkaline pH of the gut, which solubilize the crystal. The activated toxin bind to the surface of the midgut epithelial cell and create pore that cause cell swelling and lysis and eventually cause the death of the insect. Then specific Bt toxin genes were isolated from this Bacillus thuringiensis and incorporated into several crop plants such as cotton. So the choice of the gene depend upon the crop and the targeted pest. As most Bt toxin are insect group specific, the toxin is coded by a gene CRY-AC name CRY. So there are a number of them. For example, protein encoding the gene CRY-AC and CRY-2AB control the cotton bollworms. Then the CRY-AB control the corn borer. Then pest resistance plant. Several nematodes, parasites or wide variety of plant and animal include human beings. A nematode, Melediogen Echinoctia infection, the root of tobacco plant, and cause a great reduction in the yield. A novel strategy was adopted to prevent this infestation, which was based on the process of RNA interphase. RNAi takes place in an eukaryote. So what is this RNAi? RNAi is a gene slicing or gene knockdown technology. It works at RNA level. It targets the organism where there is an mRNA sequence. and interferes with the protein synthesis. This RNA interface acts as a mode of gene regulation and work as a defense mechanism against any pathogenic RNA, any exogenic pathogenic RNA. So its work is to protect, and it works as a defense mechanism against any exogenic pathogen. Organism is a method of cellular defense, and this method involves silencing of specific mRNA due to complementary dsRNA molecule that bind and prevent the translation of slicing mRNA. The source of this complementary RNA could be from an infection by virus having RNA genome or mobile genetic element transposons that replicate via an RNA intermediate. Using agrobacterium vector, nematode-specific genes were introduced in the host plant. The introduction of this DNA was such that it produced both sense and antisense RNA in the host cell. These two RNA, being complementary to each other, form a double-stranded dsRNA that initiated RNAi and thus sliced the specific mRNA of the nematode. This consequence was the parasite could not survive in a transgenic host expressing specific interfering RNA. So this transgenic host expressing specific interferic RNA, the transgenic plant therefore got itself protected from the parasite. Now this was all about the application in the agriculture. Now application in medicine. So the recombinant DNA technology process have made immense impact in the area of health and healthcare by enabling mass production of safe and more effective therapeutical drugs. Recombinant therapy do not induce unwanted immunological responses is common in case of similar product isolated from non-human sources. At present, 30 recombinant therapeuticals have been approved for human use worldwide. So, genetic engineering insulin. Management of adult onset diabetes is possible by taking insulin at regular time interval. Now, if you discuss this, you would soon realize that one would have to isolate and use insulin from other animal. So, insulin is isolated from other animal, be just as effective as it is secreted by the human body itself, and would it not elicit an immune response in the human body? Now imagine if a bacterium were available that could make human insulin. Suddenly the whole process becomes so simple. You can easily grow a large quantity of a bacteria and make as much as insulin as you need. So think about where insulin can be orally administered to diabetic people or not. Why? Insulin used for diabetes was earlier extracted from pancreas from a cattle or pig. The insulin from an animal source could cause some patient to develop an allergy or other type of reaction to the foreign protein. Insulin consists of two short polypeptide chain, the chain A and chain B, that are linked together by a disulfide bridge. So in mammal, including human, insulin is synthesized as a pro-hormone, like a pro-enzyme. The pro-hormone also need to be processed before it become a fully mature and functional hormone, which contain an extra stretch called the C-peptide. The C-peptide is not present in a mature insulin and is removed during the maturation in insulin. The main changes for the production of insulin using rDNA technique was getting insulin assembled into a mature form. So in 1983, Eli Lilly, an American company, prepared two DNA sequences corresponding to A and B chain of human insulin and introduced them in a plasmid of E.coli to produce the insulin chain. The chain A and B were produced separately, extracted and combined by creating disulfide bond to form the human insulin. So here we can see the diagram, maturation of the proinsulin and insulin in a simplified way. Then gene therapy. If a person is born with a hereditary disease, can a corrective therapy be taken for such disease? So gene therapy is an attempt to do this. It is a method that allows correction of the gene defect that has been diagnosed in a child embryo. The gene are inserted into a person's cell or a tissue to treat the disease. So what is the role of this gene therapy? To correct the deficiency, to correct the mutations inside that human body which are involved for a normal gene into an individual or an embryo to take over the function of a non-functional gene. So the first clinical gene therapy was given in 1990 to a 4-year-old girl with adenosine deaminase ADA deficiency. So this enzyme is important for the immune system to function. The disorder is caused due to the deletion of the gene for adenosine deaminase. In some children, ADA deficiency can be cured by bone marrow transplant. In other, it can be treated by enzyme replacement therapy, in which functional ADA is given to the patient by injection. But the problem with both of these approaches that they are not completely curative. At a first step toward gene therapy, lymphocytes from the blood of the patient are grown in a culture outside the body. A functional ADA cDNA used as a retroviral vector is then introduced into these lymphocytes, which are subsequently returned to the patient. However, as these cells are not immortal, the patient requires periodic infusion of such genetically engineered lymphocyte. If the gene is isolated from mature marrow cells, producing ADA is introduced in the cell at an early embryonic stage, it could be permanently cured. Then next point in NCERT is molecular diagnosis. We know that earlier diagnosis and using in pathophysiology is very important. So serum, urine analysis, etc. Our earlier detection is not possible here given the method of diagnosis. So in recombinant DNA technology, polymerase chain reaction, and ELISA, that is enzyme linked immunosorbitol assay, these are some of the techniques that serve the purpose of earlier diagnosis. So it can detect the presence of the pathogen or bacteria or virus in normally susceptible genes. So when in the pathogen is already very high in the body, so very low concentration of the bacteria or virus at the time when the symptoms of disease are not yet visible can be detected by amplification of this nucleic acid by PCR. Now I make a video on PCR. You can watch the video on PCR short, in which I have given what is polymerase chain reaction and how it works. So now moving on to the topic, it is used to detect HIV. On HIV also I have made a video, you can watch that. It is susceptible to the AIDS patient and it being used to detect mutation in gene in susceptible cancer patient. So it is powerful technique to identify many other genetic disorder. A single strand DNA or RNA tagged with a radioactive molecule is allowed to hybridize its complementary DNA in a clone or cell by detecting using autoradiography. So clone have been mutated gene which hence not appear in the photographic film because the probe will not have complement with the mutant gene. So ELISA is based on the principle of antigen antibody interaction. Infected by the pathogen can be detected by the presence of antigen, protein, glycoprotein, etc., or by detecting the antibodies synthesizing against the pathogen. So transgenic animal. Now next topic is transgenic animal. Animal that have DNA manipulated to possess and express the extra foreign gene are known as transgenic animal. So transgenic rat, rabbit, pig, sheep, or whatever animals they are, fish, have been produced. So how they are presumed DNA is manipulated to possess means there are some changes made in the DNA or the foreign gene for making it a transgenic animal. So normal development, this can be specifically designed to study how the genes are regulated, how they affect the normal body and its development, how it makes the changes inside the body in development, to study the complex factor which are involved in insulin like growth factor. Insulin is important because it was produced in the animal. So by introducing gene from other species that alter the formation of this factor and its biological effect, what are the effects of this gene, the biological role of this factor in the body is important. Then we have to study the disease. So many transgenic animals are designed to increase and understand how a gene is developed and it causes the disease. So these are specially made to serve as a model for human disease, so that investigation of new treatment, we can find out new treatments for the diseases and making possible by that this transgenic model which exists for many human diseases such as cancer, cyst fibrosis, arthritis and Alzheimer. So biological product, medicine required to treat certain human diseases can control biological product, but these products are expensive. So these transgenic animal produce useful biological product, a portion of DNA or a gene that code for a particular product, for example human protein. In human protein, to treat any disease, for example here we have taken a disease treatment used to treat epicema. For that a human protein alpha 1 antitrypsin is required. Similar attempts are being made for PKU, that is the phenylketonuria or cyst fibrosa. So there is a specific human protein required to treat a particular disease, and this have been made for the treatment of various diseases in 1997. The first transgenic cow, Rosie, produced human protein-enriched milk, that is 2.4 gram per liter, and the milk contained the human alpha-lactalbumin, and which was nutritionally a more balanced product for human babies and than the natural cows. So these are made to make good productions of the various biological product. So transgenic animal are useful for making biological product. Then vaccine safety, transgenic mice are being developed for using we can test or inject a mice for testing for the safety of vaccine before we use it to the human beings. So transgenic mice are being used to test the safety of a polio vaccine, if successful and was found to be reliable, and that could replace the use of monkey to test the safety or a batches of vaccine. Now chemical safety testing, it is known as toxicity or safety test. So the product in the same as that for using for toxicity of the drug, transgenic animal are made that carry gene which them more sensitive to toxic substance than non-transgenic animal. They are exposed to this toxic substance and the effect is studied. Now toxicity test is such animal will allow us to obtain result in less time. Then ethical issues. Manipulation of living organism by human race cannot go any further without regulation. There are some ethical standards that are required evaluate and morality of all human activity that might help or harm the living organism. So the biological significance of such thing is important. So genetic modification of organism can have unpredictable result when such organisms are introduced in the ecosystem. Therefore, Genetic Engineering Approval Committee, the Indian government has set up an organization and which will make a decision regarding the validity of GM research and safety introducing the GM organism for public services. So the modification usage of living organism for public services as food and medicine sources, for example, has also created problems with the patent granted for the same. So there is a growing public anger that certain companies are being granted patent for product and technology that make use of the genetic material, and because of that they have been identified, developed and used by the farmer and indigenous specimens. So the originality of the plant or species is being disrupted because of these genetically modified crops. So original crops are, instead of the original varieties, these genetically modified crops have been made. So rice is an important food and the presence of which goes back thousands of years in Asian agriculture history. Here are an estimate of 20,000 varieties of rice in India, and this diversity of rice in India is one of the richest in the world. So basmati rice is distinct for its unique aroma and flavor, and 27 documented varieties of basmati are grown in India. There is a reference to basmati in ancient texts also in poetry, as it has been grown for centuries. In 1997, an American company got a patent right on basmati rice through the US Patent and Trademark Office. So this allowed the company to sell a new variety of basmati. in the US and abroad, and this new variety of basmati has actually been derived from the Indian farmer varieties. So Indian basmati was crossed with the semi-drop variety and claimed as an invention or a novelty. So the patent extended to functional equivalent, implying the other people selling the basmati rice could be restricted by the patent. Several attempts have been made for the patent use, production and processing based on the Indian traditional herbal medicines. Example: turmeric, neem. If we are not vigilant and we do not immediately counter these patent applications, our country individual may encash our rich legacy and we may not be able to do anything about it. So biopiracy is the term we refer to use bioresources by multinational companies and other organizations without proper authorization from the country and people concerned with the compensatory payment. So most of the industrial nations are rich financially but poor in biodiversity and traditional knowledge. In contrast, this developing and knowledge related to bioresources, traditional knowledge related to bioresources can be exploited to develop modern application and can be used to save time, effort and expenditure during the commercialization. This has been growing realization of the injustice in adequate compensation and benefit sharing between the developed and developing countries. Therefore, some nations are developing laws to prevent such unauthorized explorations of these bioresources and traditional knowledge. The Indian Parliament has recently cleared the second amendment of Indian Patent Bill that takes such as issue in considering the terms and of the emergency provinces researches and developing initiative. So this was all about the chapter of biotechnology and applications. So it has given a human several useful products by using micro, plant, animal and the metabolic machinery. So important points, we know that tissue culture, PCR, then recombinant DNA technology, then genetic modified organism. These are important in this chapter. Various gene therapy, transgenic animal, etc. And what is ADA deficiency? Thanks for watching. Do like, share and subscribe.